It's only yeah, like
So we continue with Professor Dr. Mahmut Cerkez.
Hello again. Good afternoon. Yeah, I'm Mahmut. I work as a healthcare scientist in the genetic department in the hospital, yes, University Hospital. And I also have a new establishment called ICODAAS in Cyprus. So I'm a business partner of the medical center. Today, I changed the title because I thought it would be more fancy to call it the way that are we really born to do smoke or drink. I'm not going to talk about the drug abuse. I'm going to mention the tobacco consumption and also drinking. Yeah, I asked the artificial AI to draw my the title, and while I was preparing, I had a call from my friend, and she was saying, "What you're doing?" I said, "I'm actually preparing my talk for the conference," and she said about like drug addiction and alcohol and whatever. And then she said, "What are you doing?" And I said, "I'm having a whiskey." So she suggested me to share the photo. But this is the photo that I sent to her. But actually, yeah. So I don't have any conflict of interest while I'm related to my talk. So I want to give a little bit of information about the genetics and the molecular biology because I think because we have different backgrounds here, so or maybe I'm trying to remind you the basic biology. The central dogma of the molecular biology is very important and crucial. And we have the DNA molecule, which is it has our knowledge from generations to generations. And DNA is the only molecule that can replicate itself, but it is inside the nucleus, so it needs an internal protein, internal molecule called RNA, to move the information from the nucleus to the cytoplasm, and it goes to the ribosomes to make meaningful sentences. Before digging into that puzzle, what we know, and I have to mention here, and there are a lot of women here. This is the photo of the first photo of the DNA showing double-strand break, double-stranded molecule, and it has been taken in 1952 by Rosalind Franklin's PhD student, but they didn't believe her because she was a woman, and James Watson, which has passed away recently, and Francis Collins discovered a year later, and they believed them. So, but I always mention the Rosalind Franklin. I do have 3 cats, Watson, Crick, and Rosalind. So after 40 years, actually, like we found the DNA molecule, they discovered and they explained how the DNA molecule is. They got the Nobel in 1962. She couldn't get the prize, even she won, but she passed away due to the cancer back then. So what we did after the 40 years, we managed to, we scientists, we managed to took the nucleus, took the DNA from the nucleus and sequenced them. But it took 13 years, the Human Genome Project, from 1990 to 2003. And what we know now, there are 3 billion letters, 3 billion base pairs, or 6 billion letters, and approximately we have 22,000 of genes. But this was only the beginning of our chapter. So genetics is a very new field. So we are, I think in the past 15, 20 years, that we are using the knowledge of the genetics. This is very recent. So, as I said, it took 13 years to complete the human genome. Now we can do in our laboratories in 1 week, we can actually sequence the genome. So, what we know, we do have identity 99.9% similarity. There's very small differences, 0.01, but it looks like it makes us very different. So we have our close relative Neanderthals. So we do have similar DNA 99.7%, and the chimps, our common ancestors, 96%. But we shouldn't forget that 50% is similar with the banana. So, like how So after that, we start to know about the genetic changes. We call them polymorphism, genetic variations. So, and then the genome-wide associated studies was born. So we start to use the knowledge or to try to associate the genetic variations with the disorders or behaviors. And this was the project that I was involved when I was in the UK. So there was the first it was 4 population, then they increased it to the 26 population to check the differentiation among the population. And we know that they will all come from Africa. So the most genetic variations they are in Africa. So the human genome, only the small percent of the human genome is decoding the proteins. So 1.5%. So the rest is a gap DNA, but actually they are retrotransposon, which they are coming from the RNA, the viruses, RNA viruses, integrating our genome, and also the other repeated DNAs and other tandem repeat the DNAs. But that small changes can be related with the disorders, but generally the genetic variations, they can create the diversity. So what does it mean? This is the very small, that the beta globin gene is a very small gene, and it, our red blood cells, they do have globin gene, alpha and beta. So I put this because it's the Mediterranean, thalassemia is a Mediterranean disorder, Mediterranean-related disorder. So it's very common here in the Cyprus and the Turkey, like the Mediterranean basin. So small changes like this one, if you look at here, the A, if it changes to the T, it can cause thalassemia or sickle cell. So we know that the African descendants, they do have huge frequency to carrying the sickle cell. So what does it mean? For example, Jenny, again, alcohol. I just realized it now. So Jane wants to get a beer. So if the one letter changes to the A, that means Jane wants to get a bear. Completely different sound. Or if you get more mutations, it's Jane wants to get a beard. So it's completely different. So these small changes, they can cause monogenic disorders. The first patient is our patient, Ella. She has Whitman-Merton syndrome, so progeria type syndrome, and Cerkez Holborough syndrome, thalassemia, phalangeal syndrome, and Marfan syndrome. So they are all unique rare disorders, but only one letter is enough to have the disease. However, when we say genetic variations, for example, Jane wants to get a beer again. Jane wants to drink a beer. So different way to say, but the concept is the same. Or Jane wants to buy a beer. So this is the diversity. So variations, they can create diversity. Why do we care about genetic variation? Because we know that it can show us the phenotypic differences. Also, genetic variations has a predisposition to complex disorders like obesity or response to the drugs and environmental factors. We know that there is for example, caffeine or the blood thinner that we give to the patient who has the MI or embolia, the background. We give them blood thinner, so we know that each patient has a different the catabolism system. So we have to check the genetic variations. They can change the response to the drugs. Also, genetic variations help us to go back to the ancestral human migration story, so we can check, we can get information about the human ancestral DNA. So, if you look at the tobacco and alcohol use, which is the two most common consumption globally, so in total there are 11 million deaths per year related to the tobacco and alcohol use. Even there are lots of prevention programs, still the alcohol and the tobacco is the one of the main cause of the deaths globally. This is I took this graph from the WHO's the website, so they have a prediction in 2030. Still, it will be the lead, but it will get down. But we know that undeveloped countries or developing countries, they do have more consumption of the tobacco. So is it nature or nurture? So if you come into my title, are we born to smoke or the drink? Actually, this comes from the twin studies. They do check the monozygotic and the dizygotic twins, and it shows that the smoking initiation, it has a link with the genetic 40-50%. Always there's environmental factor effect too. But nicotine dependence has more genetic association. Again, alcohol use, again, is like between 40 and 60% associated with the genetic variations. So there's the biggest study ever in 2022 by Sanders and colleagues. So they checked to 3.5 million people, and they associated with the 4,000 genetic variations, genetic markers. And to sum up, the first gene that you can see, CHRNA5A, is related with the nicotine use. So the one variation there, one single letter, the letter changes, and there's a link to the ethanol consumption. The two genes are related, and serotonin transporter gene is related with the behavioral, especially for the schizophrenia, bipolar predisposition, and also in our study we showed that it's related with the serotonin, the tobacco, sorry. So this was the biggest ancestral study ever, but the study was lack of the global data. So 78% of the participants they had a European descent. So there wasn't any Middle Eastern data in it. So, and I had to give an interview to the Nature about it. They found me the Miriam by that time. So we mentioned that the Middle Eastern populations, because of the cultural effect, there's a huge use of the tobacco, shisha, or like water pipe, whatever. So and then we gave our study to the example. But as I said, there was a huge study. They checked all the population, but there wasn't any Middle Eastern. So it can show us the good European background, European people from the European descent can be good related, but there wasn't. They had a huge the gap about the Asian and the Middle Eastern population. So what we have done, we published this study. We checked the smokers, non-smokers, and ex-smokers, which they have stopped 5 years ago. So it was as you know, North Cyprus is a very small population. Turkey and Cyprus are a very unique island population. There are a lot of island-specific mutations and genetic variations that are specific for the island. So we first check there wasn't any difference between the sex, and also we did check some parameters like biochemical parameters, and also BMI for the mass index. So we didn't find any difference between non-smokers, smokers, and ex-smokers. However, we checked the serotonin gene, and serotonin gene in one there's 3 variations of the one polymorphism. So we have checked the genetic variation association in Turkish Cypriot population for that serotonin gene. And we didn't find any variation of this STIN9 variation. I'm going to show you here just a second. Yeah, it's here. So we didn't find any STIN 2.9 variation in interdisciplinary population. So mainly it was 2.12 and 2.10. So if you go back, so there was a statistical difference between smokers and non-smokers for the genetic variation in the serotonin gene. And we compared that smokers, non-smokers, smokers, ex-smokers. We found the statistical difference also, and smokers combined with the non-smokers or ex-smokers. So what does it mean? It means that if you have 2.12 variation, you are more willing to smoke because their serotonin is disrupted. So you need to get the nicotine from the outside. But if you have 2.10 variation, because your serotonin is not that disrupted, it's a gene, it's a protein, so you don't need the nicotine to take from the outside. So which we have noticed in the ex-smokers. And so this was like the one country-based study, of course, but we know that the serotonin also has a association with the major depressive disorder and bipolar disorders. So to sum up, if you merge all the information from the genome-wide association studies, these are the nucleotides. So nucleotide changes in different genes can actually explain or associated with the nicotine and alcohol use. For example, the first one is it was Saunders' study, so it's related to nicotine dependence. And what we found in interdisciplinary that the serotonin gene mutation or variation can increase the smoking consumption or tobacco consumption. So, what does it mean? How we can use them in our clinics, or how we can help the patients with that. We know that when we were born, our genetic information is there. So we can, nowadays in the developed countries, I know that they do start the genome sequencing, but what we can do to prevent it for early intervention, we can use them as a pharmacogenetic approaches, which to check the genetic variations and can help, we can prevent the drug or medicine use, or also we can check for the late onset disorders, this information to give to the patient with it, of course, with the counseling to prevent their future disorders such as Alzheimer, Huntington, or maybe cancer. So what we know that the variation in the genes, they do have efficiency on different medicines also. So what we know, the personalized medicine is improving, and personalized diet, personalized medicine, so precision on medicine is important nowadays. So we have to use this information to prevent the people from getting disorders, diseases, and also having them a healthy life, a long, healthy life. So it comes up with the longevity. So it's the science of living longer. So we know that 25% genetic factors are important for us to get a good, healthy, long life. But we know that the main factor is lifestyle and environment. So if you combine them, so 75% is lifestyle and environment. So genetic has a very small role in it. But we know that also nowadays epigenetic approaches, which is the modifying genes, and the microbiome in our gut also is important for us to have long and healthy life. So, as I said at the beginning, it's very early nowadays. The genetic is very early. To know the genetic data is very early. It's after 2003, so 20 years old. Using all the genetics studies and using this information in the medicine. We know that the behavior from that genetic associated behavior, and it's important for us also to prevent the disorders and giving personalized guidelines. So what I try to say is personalized therapies maybe according to genetic information or using the genetic information, it will be more useful in the future to help patients more. So, the conclusion, I took off the Middle Eastern here. So it's a huge gap, as you see. So they have done good studies, but most of the studies in developed countries are related with the European descent. So Middle Eastern or Asia is always lack of from that studies. So maybe we have to encourage or we have to share more data internationally. So national genetic database are important. I think Turkey just launched last year genetic variation database. When I was in the UK, like 20 years ago, when I was studying, it was 2005. I think it was 2005 or 4. They had the UK biobank already. So as a region, we are coming a little bit behind, but it's important to our studies to contribute to the global literature, I think. So I'm happy to take if you have any questions. If you don't have it, it's okay. Okay, thank you very much.
Oh, wait. They're thinking. Go ahead, Eva.
I think it was a fascinating talk. Thank you so much. Not boring, not a single second. And I just wonder about the link, genetic variation with the effect on serotonin and nicotine dependence and impulsivity. Could you say a few more sentences? Is it
Yeah.
Is it valid for all parts of the world? Or?
Well, actually, what we found we didn't have the one specific variation in Turkey, Cyprus. This the cohort that we studied at least. But we know that the Europeans, they do have that variation more common than us. Again, it's the low frequency, but they do have it. So what we concluded that the use of the smoking, the tobacco, it will be based on the cigarette users. They have another addition to the group, so we don't know. So you have a good point here. So we only related with the tobacco use. It was one of my students' project actually. So we related to the tobacco use, but maybe in the behind they have another addiction too. So what we compare with this using the tobacco with that specific variation, if you need more nicotine to stimulate your serotonin level.
Is it driven by bad mood then?
Well, it can be, but we didn't find that variation in the smokers. So that's why we came up with the idea that if you are a smoker and you have the variation, it's in your genes that you are willing to have some supplement or some additional like tobacco, additional stuff to stimulate your serotonin levels. Yes.
Very good presentation. I really enjoyed it. Not my area of expertise, but it was very interesting. I learned a lot. I just wondered what your future research plans
Well,
are. I am
a very busy scientist actually. So I research I started with population genetics, and then last 10 years I focus on the rare disorders. And so this is like the genome-wide study, like to check from the data that we had. But generally, I am interested in the translational medicine to find the therapies for the rare disorders. And mainly the Cyprus population, but in general, I'm trying to discover the new genes actually, and to understand their function using the animal models to help find the therapies for the various disorders, which most of them they affected. The children are affected, more than 50%, and most of them they die very early age. So this is my main focus. So I am not doing this stuff anymore.
Do you have one, Kayla? No, sorry. So, in 25 years, do you think we'll all have our own personal genome done? But then there's so many ethical questions about, I mean, it may really help us with disease, and we're certainly doing it with cancer now and using that. But, I think drug abuse would be an area where it could be very helpful for prevention. But do you see then worry about, should we send this child to school, the IQ's lower, those kinds of things?
Yeah, you're 100% right, and especially the insurance problem as well. Yeah. So, and we had that problem for 23andMe. You know that there was an American company, and they were checking if you are predisposed for the disorders, or like you get the ancestral history. But what they have done, they sold all the data, and they share people's information. So, well, it's difficult to say, but I think in New Haven, Connecticut, they started to do and in UK now, they have, for each born baby, they do sequence.
Really?
Yeah, from the
Wow.
from the heel blood.
Yes.
And they do sequence them. And so the important thing is how we're going to use the data and how we're going to save, because it has your own knowledge, which you don't want to share with your family. So we do have cases like, for example, Huntington patient, or the family, they lost their father from Huntington, and they're twins, identical twins. So one of them, he wants to learn if he's going to have a Huntington in the future, and other one he doesn't want to learn. But it's because they do share the same DNA, but they have they are two different personalities. And I think this was the case that went to the court also, and they decided to tell if you want to know it's your information, so you have to know. But yeah, I understand your concern, and I think it's very right. I don't know, to be honest, how, for example, the insurance companies in the States, they want it. Also, sometimes we do test pregnant ladies when they have the 10-week of gestation. We do check. There's a screening test that we check. We took the mother's blood, and we check the baby's DNA that are circulating in the mother's the blood system. So we are checking if there's Down syndrome, just a screen, like some chromosome abnormalities. But we do get information about the mother too. Yeah. So, and we can check. There are companies nowadays, they check if you have a breast cancer predisposition, Angelina Jolie, like BRCA1 and BRCA2. And so, do we have to tell the mother to be, because she's already pregnant? She has another enjoyment in her life, and others and also we have to tell her, oh, you're gonna have a cancer, or you might be willing to have a cancer. So these are still ethical considerations, and I think we have to, as a scientist, or maybe the big community, the guidelines has to be very clear about what we do see. For example, in my practice, we don't report any late onset disorders genetically, and when we have a child, we check mother, father, and children, or child. And sometimes we come up that the mother or father is not biological. But we don't say anything to them, because we don't want to destroy their life, or especially the child will get affected more. We do see but we don't say unless they ask for us to check.
One more, just little one, but you know, you brought up about sharing data. And if you get more worldwide data, you might increase the statistical ability to identify things. But on the other hand, if you have something in a small community like the Turkish Cypriot community, and it gets diluted in a giant database, you may lose the
You're right.
value of the data that you have.
Yeah, you're right. That's why I'm saying we need to have national database,
Yeah.
but also we have to share the information
Do both.
for the researchers. I will give a small the example for that one. There was I think 2-3 years ago, FDA approved one of the lung cancer chemotherapy, serotonin, I don't remember now, it starts with the S. Then immediately when I saw, I just went back and checked there was a specific mutation in the KRAS gene. The KRAS gene mutation, we know that is very progressive. So the chemotherapies are very resistant to the chemotherapy. So there's a specific mutation there has effect, has a good side of effect for the patient. So what I did, back then, I went there and checked all my lung cancer patients who are alive. And there were 12 patients alive at the time. 11 of them, they were Turkish Cypriot without having the mutation. One of them had the mutation for this drug, and he was British. So, because we do share the European or UK data or States data, but we don't share our data, which is important for the researchers because they're going to check, this is a very frequent question. That's a very frequent mutation. So let's make a drug or design a drug for that mutation. Yeah. That's why it's important globally and also nationally. Yeah.
Thank you.
Thank you very much. Cheers.
Okay, hello again. I will try to summarize you some of the STI, sexual transmitted diseases, infections, especially the risk groups and the substance using populations. Yes, we know that irregularly all over the world, Trichomonas vaginalis, Chlamydia trachomatis, Neisseria gonorrhea, and the Trichomonas pallidum is the STI, sexual transmitted infections. And especially we see these infections, substance using populations. And at the end of my presentation, I'm trying to talk about some prevention strategies, that how can we stop these infections. We know that this group is going to include the bacterial, fungal, viral, and the protozoan infections. And they're going to be had sexual contact especially. And we are going to be mostly observe this infection all over the world. Increased risk of this advanced group. What the Hugh said? Hugh said that it's a global burden of the major sexual transmitted infections. Especially you can see the people with the herpes simplex virus and the new infection in 2020, the STI. This STI is going to include the chlamydia, gonorrhea, syphilis, and the trichomoniasis. So we will still fight, we will still the global burden about these infections all over the world. And of course, you can see the percentage of the infections. This is Trichomonas vaginalis, it's 42%, and we have the Chlamydia trachomatis, it's at 34%, and 22% is the Neisseria gonorrhea when we talk about the STI. What about the reports, the research? What's going to be tell us? The Shannon Seal and et al. From the United States reported that 20 million new STI infections in their countries, and we have seen similar data in the lab area. They have reported 25% of the chlamydia infections, the younger than 20 years old. This is very, very critical. In the Iran, there is another research. You can see that we had see these infections in the adolescent in the 2025 per 100,000 persons 8 years. And another infection in the reports in the Italy, we see that increase, especially the age between 15 to 24 years old, infection in the last 20 years. Yes, Trichomonas vaginalis This is the parasitic infection. We call them the infection is trichomoniasis. And another microbiological infection, we're going to be call them neglected sexual transmitted infections or the silent sexual transmitted infections. Yes, it is treatable and curable. And we have that there's a new cases in 156 million. And the age group is starting with 15 to 50 years old. And female especially, they're gonna have this vaginal discharge and they're gonna have a risk of the pelvic inflammatory disease as well. And we're gonna be associated with this infection, especially here, HIV acquisitions. Yes, what's the transmission? Yeah, we know that sexual contact, mother to child, and the contact object is the dominant transmission way. And we have the symptoms, both of the disease infection, men and the female as well. And in the women, we see the vaginal discharge, the twitching, the unusual odor, and the greenish and yellowish fluid. And the men, yes, this is the critical, usually asymptomatic. The man is going to have this infection, but not any symptoms. So they're going to be spread to the infection to the women or the another partners. That is the critical way why we see these infections all over the world. Because most of the STI infection is the asymptomatic. Yes, how can we diagnose in the medical microbiology lab? We are going to use the different type of the specimens. We're going to use the urine, blood, the cervical or the vaginal swab samples. Yes, we have the conventional methods like the staining methods under the light microscopy. We can easily see this infection. Yes, if you have ability, we can culture as well. These are the gold standard in medical microbiology. And yes, we have the new technology like the Mahmut is going to be mentioned there. We are going to use the molecular techniques like 2D imaging or the nucleic acid testing, and they're going to help us. And another important thing that we have the point of care test as well. If you have anything that you are going to be infected, you're going to be usually take the not going to chip. You're going to take the point of care test, and you can make the diagnosis. Can we treat? Yes, we can treat. We suggest the metronidazole, for example, and the antibiotics or the antiparasitic infections and drugs or can be treated. But the main thing is if you are going to be taking this infection, it's asymptomatic, you haven't any symptoms, and you do not get started to treat, but you can still spread these infections. Yes, when you are going to make the research about the world, we can find some articles that I'm going to want to show you. For example, this is systemic review and meta-analysis of the global prevalence and infection risk of vaginalis. They're going to indicate that the forest plot shows that there was moderate variation between the drug group and the control group, and they're going to find statistically significant effect about these infections, about risk group of you can use these drugs. And this is another example. It's going to be a study. They're going to be studying the 584 peoples, and the prevalence of the trichomoniasis is 43.6%. They have past month substance use was high, with about 90 of the women reporting at least daily drug injection. Half reporting past month use of methamphetamine, 19% reporting alcohol use with clients, and 58% indicate drug use before or during sex with a client. And this is very important. Less than 150 of participants had ever had a genealogical checkup, and the prevalence of STI was high overall. That is the very important thing why we are not going to stop these infections. Chlamydia trachomatis, this is a bacterial infection. Yeah, we know that estimated the 128.5 million new infections, and the age group is going to be similar. Yes, we can treat, we can use antibiotics or treat these infections. And however, if you are not going to be treating this infection, we have the serious problems all of the life of these patients. This is transmission growth. You can see that mostly the percentage is the vaginal intercourse, anal intercourse, oral sex, and mother to child. But predominantly, we can see to the vaginal intercourse about this transmission of this infection. Yes, this is the symptoms. Pain during urination, fever, abdominal discharge, vaginal effusion, penile discharge, and the painful intercourse for these patients infected. How can we diagnose? Yes, we're going to be using rapid test, antigen-based test in the lab. And sometimes we're going to use the nucleic acid test as well. And if we have the ability, we can culture these microorganisms into lab as well. And we can see it under the microscopy. What's the differences about these tests? You know that some tests is the clinical microbiology, they have different sensitivity and specificity. That is the one important thing. And another important thing that where you're going to live. You're going to live developing countries or the undeveloping countries. Because the money is also bit tall in this diagnosis as well. That is also be very, very changed according to the countries. Can we treat? Yes, we can treat. Uncomplicated chlamydia is treated with the antibiotic tablets, including doxycycline or the azithromycin. Yes, repeated infections can occur if sexual partners are not treated, and if individuals have sex without condoms with someone who has the infection. The important thing that they should notify their sexual partners to get tested and treated. That is the important thing. Because the patient is going to come, they're going to make the diagnosis, and they set the patient that please tell this situation to your partners. Okay, the partners have to come to the check, but lots of them is not going to go and tell them their partners about these infections. And the infection is going to be spread. What about the research area? Yes, we know that this is the good example, the prevalence and factors associated with sexual transmitted infections among people who inject drugs in Santiago and Tijuana border region. They're going to be report that 16 of 519 participants tested positive for urogenital chlamydia, 1.2% tested positive for urogenital gonorrhea, and 3.5 tested positive for syphilis. And there were several significant differences between participants who tested positive for bacterial STI and those who did not. Of course, the HIV, when you look about the table, you can see that HIV is going to be much higher. But when we look about the STI infections, we can see that lots of them totally have the STI infections. And they also make this table like this. They're going to be make demographic, some information about the patients. And they were significantly higher also having the bacterial STI among participants who reported recent stimulant use, those who reported having previously been diagnosed with the bacterium, and participants who reported recent methamphetamine use had 2.6 to 1 times higher also of any bacterial STI compared with those who did not. And no other substances were significantly associated higher also having a bacterial STI. So that means that the risk group of this is going to be related strongly with the STI infections. This is another example. They're going to be doing in the Tokyo between 2014 and 2022. This is the retrospective cohort study. Yes, the syphilis was the common infections, and in the total, 142 patients included 7 heterosexual males, 170 non-inject drug use, man-sex man, and 18 man-sex man-inject drug use, experienced 213 syphilis infections. 55 patients had multiple infections during this study period. This is very common about this patient that we can see the multi-common infections. And by the end of the observation period, 19 patients had been infected with syphilis, only once in their lives, and the others experienced syphilis infection. This is another example. They're going to be having testing for the sexual transmitted infections for persons who inject drug hospitalized with serious injection-related infections. They're going to total 37 unique patients positive, at least one STI infections, and testing remained lower among men compared with the women. Yes, when we look about the world, we can see that most of the infection STI is predominantly, unfortunately, women. And they got all patients receive treatment for the STI during their inpatient admissions. Next, Ergören is the another STI infections. They estimated that 82.4 million new infections among adults globally. Most women with gonorrhea don't have symptoms. This is another thing, it's called asymptomatic. And vaginal discharge is going to be common. While most men present with discharge from their penis, if we can treat these infections, but if left untreated, they're going to have infertility in the both and the men and the women. Yes, the transmission way, sexual contact, injection drug use, mother-to-child transmission, and the contacts. Symptoms is going to be similar to the other STI infections: pain or burning, vaginal bleeding, painful sore or swollen testes. And the pictures is under the microscopy, easily just understanding with the vaginal swab. You can see the gram-negative record, and we're gonna make the diagnosis in 5, 10, and the 15 minutes. Yes, this is the classical method, microscopic slide. We're gonna stain it. If we have the chance, we also will make the contours as well. We can use the molecular biology test, and we can make the identification. We're going to do the antimicrobial testing as well, and we are ability to chance to make the diagnosis. Can we treat? Yes, we can treat these infections. Treat with antibiotics like cephalosporin, for example. But another important thing that they should notify their sexual partners to get tested and treated. This is the I think important thing about this one. What about the research tell us? This is the Mahmut et al. Studies, and they're going to be reported that about 3.6% of respondents report having an STI in the past. Of who that had STI in the past year, majority were 22 and 25 years old, female, non-Hispanic white, never been married, reside in a metro area, and had health insurance. And 45.7% had some college, associated degree, 71 and 1% had full-time, part-time employment, and 44 and 3% had an annual household income of $20,000 between the $50,000 restaurant. Triple pallidum, yes, this is another important STI infections. Again, we can see the asymptomatic infections in here, and who estimated 8 million adults between 15 and 49 years old, occurs in 2022. And we can treat as well, but the key population is especially gay men and other men who have sex with men are disproportionately affected. Yes, it's a similar sexual contact, mother to child, blood transfusion sometimes, and injection drug use is the transmission of these infections. Sore thorax, skin rash, genital sores, pain, urination, these regularly symptoms that we can see in these patients. Yes, we have diagnostic chart here. You can see as well, we are going to use the dark field or the direct fluorescence antibody test, but normally we're going to first start with this screening test. And if it's going to be positive, we are going to do the confirmation about another test of using the patient's serum. And we're going to try to make the diagnosis about these patients. Can we treat? Yes, we can. We're going to use penicillin, and we're going to treat these infections. The problem is not going to be treated. The problem is going to make the correct time the diagnosis about these patients. This is the ECDC report in the world. You can see that the brown the countries is going to be higher than 7.0 notification rate about these infections. And this is another table between the ECDC as well for the symptoms. And when you're going to look about the years, we see that not 6 months person is going to be the incidence is going to be higher about these infections. And what about the research? This is in the rural area of the southern China. The prevalence is the 1.2%. These factors vary by the gender. And the male group, we have significant associations with the injection drug use and the hepatitis C. And in the female group, they're going to be included schools having syphilis, ever having blood transfusion, or hepatitis B infections. So can we prevent these infections? Can we have prevent strategies, for example, in other countries or in other countries? The ECDC is going to be great, some strategies to control STI infections. They're going to be made to group. The first one is national coordination. So what we are going to do, governance, leadership, and the management partnership that we will try to do it. And strategy and action plan, monitoring and evaluation, and the program evaluation. What we are going to do in the clinical service? Yes, it's easy access to STI care. This is very, very important. So, early diagnosis. Yeah, we know that this infection is also asymptomatic. So we need time to make the symptoms. But if we have to do the diagnosis early, we're gonna be stop this infection to get it treated. Acceptable clinical and diagnosis. Effective treatment. We have to evidence-based case management guidelines, professional training of clinical and educational staff, and department management services. And another part is the public health as well. What we are going to do is health promotion and the behavioral interventions, partner notifications, outreach testing and screening program, biomedical intervention like the vaccination about some of the infections, and we need the surveillance. We need to be results. What's our population's infection? The surveillance program is not for the STI, but for the all over the world. If we want to stop some of the infections, we need to know what we are going to in our countries. This is the surveillance. And also ECDC is going to make this table as well. They're going to be think about that susceptible group, like the infections of the pathogen that we talk about it, sexual sex practice, frequency and type of sexual contact, co-infections that the pathogen has been mentioning. If we're gonna be reduce these things by using health promotion, behavioral intervention, education and training of the public about these infections, yes, we will gonna be reduced. And we have the infected group as well, like the incubation period, no symptoms, and asymptomatic infections. If we are gonna be ready to the public to access to STI care, clinical and laboratory services, department services, we also will reduce these infections. And we need to be some recovered group, like testing and early diagnosis. Yeah, it's easy to make the test or to treat, but the important thing that where is the patient? We have to be know the patient to make tests and to make the diagnosis. And effective treatment that we have a chance to treat this infection as well. Comprehensive treatment and recovered cases become susceptible again most to the STI. So we need a strategic plan for all of the countries and implementation, and we need to monitoring and evaluation about this risk group and the STI infections. So when we're gonna be talk about the loss prevention, yes, condom and lubricant distribution, needle and syringe programs, safe sex kits, and routine STI testing maybe help us to reduce these infections. Yes, my last word from the Mustafa Kemal Atatürk, because tomorrow is the 10th November. The 10th November is very, very important for us, for my countries and in Turkey. The Mustafa Kemal Atatürk is going to die on the 10th November or 1938. My last word in the English, if one day my words are in competition, it's science, choose science. And my last word in Turkish, eğer bir birbirini sözleri birinde ters düşerse bilim seçin. Thank you.
Thank you for your excellent presentation. I'm a bit shocked that there's so many cases
Yes,
and that there's this.
just a part of them, not all of them.
Yeah. Do we see a COVID effect here in the trends, or what is the reason for this increasing number of?
For the STI.
Yeah.
Yeah, they basically think this asymptomatic part. In the medical microbiology, not for especially STI, but if it's asymptomatic, it means that you get in your body the microorganism. They're going to replicate, they're going to be in your body, you're going to spread, but you don't know that you're in your body. You haven't any symptoms. That is the main critical part about this infection is going to go. Like COVID-19. For example, COVID-19 also they're going to be started asymptomatic. After the 4 and 7 days, your symptoms is going to be started. But that time, you screen these infections. That is going to be an important thing. But the main thing that I think so we have to be the good surveillance program about the risk group. For example, in the medical group of the medicine students, we are going to be a special end of the diseases when we educate them. We're going to have a list of the risk groups like the elderly, the newborn, the immunocompromised patients. But actually, basically, that time in the future, we have to add the one risk group to the substance using drugs group as well. I think so. Thank you.